Hello, for today's lesson on plant anatomy, we're going to look at three short things. Firstly, we're going to look at the basic types of plant tissue and identify what they are. Next, we're going to focus on vascular tissue in particular and look at the structures and functions of xylem, phloem and sclenchyma. And then after that, we're going to identify vascular tissues from light microscopy pictures. Normally we would do that last one in a lab, but for now we'll have to do with some pictures yourself. So firstly, let's look through a root, just to get an idea of what those plant tissues are. We have, in the very middle, we have a layer on the outside, and then we have a sort of spongy bit in between. So without going into a lot of detail, in a nutshell, the middle part contains the vascular tissue, which contains the xylem and phloem. On the outside we have the skin sort of layer containing epidermal cells that cover the outside of the plant and protect it, and then inside we've got all the other stuff really. So these could be cells that are going to photosynthesize, containing lots of chloroplasts. There could be cells that are specialized for storage of starch, and there are also a couple of cells in there that help support the plant. We're going to focus on that very top one for most of this lesson. So the vascular bundle is made of three different layers, and we'll go through each separately. We have sclenchyma, we've got phloem, and we have xylem. So let's look at each in turn. Starting with xylem, we notice that they already look like hollow straws, and they're made of hollow connected cells, although some of the ends might have perforated end walls, and this allows them to look like long hollow tubes. The outside is very thickened. They have the material called lignin in their cell walls, and this helps provide structure to hold the tubes of xylem open. However, because of all of this, they are dead cells. They have no contents, cell death has occurred inside during their formation, so they really are just the cell walls left like a husk. But the lignin in there also makes it waterproof. And from our previous lessons, we know that these are involved in transpiration. They're to do with pulling water from the roots up to the leaves through transpiration pull, with all those forces of cohesion tension by the water molecules, allowing the water to move up as a continuous column all the way from the roots up to the leaves where water is evaporating. Also, because they've got that lignin in there that helps hold them open, in a similar way you might like to think that cartilage is holding our trachea open, this provides a bit of support to the plant too. So next we have phloem, and these cells are also hollow, they're also connected, but they're filled with a little layer of cytoplasm in there too. They are alive. They have different little perforations in their end walls, which are called sieve tube plates. And these are all to do with translocation. This is the process where you're having sucrose, different sugars, amino acids and mineral ions all being brought up through the connected tubes through the cytoplasm of these living cells. And lastly we have sclenchyma. And these are dead cells again, and you can notice they look a lot more like lignin than they do like phloem, because they have that thickened outer wall with the lignin in again, and they're very, very strong to support the plant structure. And sclenchyma also makes cells and plant tissue quite useful. So you can use plants very rich in sclenchyma in industry for textiles, paper and biofuel. If you think about all of those bamboo insert product here, things that have started appearing, very useful, strong materials. So let's take a moment to review what we know and what we've learned. I'd like you to fill in this table and have a look through for the general structure. Give me a description of what it is. Tell me if it's dead or alive. Summarize the function of each vessel. What's in the cell wall? What's it made of? And is there anything inside that cell? So pause here, complete the table, and we'll go through the answers. Let's start with general structure. So as we said, xylem are hollow tubes made of many connected cells. We know that they're dead, that they transport water and a bit of mineral ions, and they're useful for a little bit of support because of that lignin in their cell wall. And inside they are hollow because they are just dead cells. Next, going through phloem. Now these are vessels, again, they're tube-like and they're made of many, many connected cells with sieve plates in between. They are living cells. You have sieve tube elements kept alive by their connections to the companion cells that carry out all their metabolic processes. and their function is to help transport dissolved carbohydrates, amino acids, and mineral ions. They do have a cell wall, but it's nothing fancy like xylem. And inside they have a bit of cytoplasm around the edge, and they have a big hollow center. Next up is sclenchyma, and their general structure is that they are separate cells joined together to form fibers. These are dead too, and they're only there for structural support like the skeleton of a plant, and they have a thick cell wall with lots of lignin, and again, like xylem, they are hollow, dead cells. So score yourself up, see how you did. Because next what we want to do is test our recall. If you can, hide your notes and try doing this without looking at anything. What we have is a picture at the bottom, and it has 3 different labels on it. We've got X, Y, and Z, and each of them is showing a different zone of the 3 things we've just talked about. What I'd like you to do is look back through our notes at the beginning and see if you can label which is which. So out of phloem, xylem, sclenchyma, what goes where? After you've done that, I want you to have a look at the right hand side, and we're going to try and draw more knowledge out by comparing the structure and function of these 2 things, xylem and sclenchyma. So there should be 2 similarities and 2 differences. Give it a go. Let's start with labeling different things. So if we look at X in the middle, we notice that they have lots and lots of small cells. If you look at the outlines, there's no particular thickening of that cell wall, so this must be phloem, the only unlignified vessel. Now we have a choice. We have lots and lots of these small cells with very big, thick cell walls, and we also have Z here, and you notice that some of them are very big. They're very big circles compared to the tissue around them. So we have a choice now between xylem and sclenchyma. So let's start with Y. Y is your sclenchyma. These are the cells that are there for structure and support. And Z, we have xylem, the vessels that contain water. So it makes sense for them to have a slightly bigger opening or diameter. So let's see what you've written for the structure and function, writing your comparisons. Our similarities are that xylem and sclenchyma are both made of dead hollow cells, and that they both have thickened lignified cell walls. Our differences are where xylem vessels are connected and can sort of transport water between neighbouring xylem vessels. Sclenchyma are separate and they form fibres running through the whole plant. Another difference is with their function. Xylem vessels transport water, whereas sclenchyma are for structural support. Lastly, let's look at a few practice exam questions. So, for our first question, we have mineral ions such as calcium nitrate and magnesium are transported in the xylem. These are mineral ions dissolved in water. What you need to do is describe how the structure of xylem vessels allows them to transport water. So how does the structure of xylem help them transport water? 2 marks. You've got 4 possible mark points you could choose from. Give it a go. So, the first thing that we need to look at is we are describing the structures. We don't need to talk about the fact they transport water with mineral ions dissolved in. That's given to you already. So all of your points should just be about what it looks like and what it's made of. So looking at our first point, they are hollow and have no cytoplasm. You could say that the vessels are open at the ends, or that these xylem vessels have no end walls. And you could also say that they have pits, or that they are strong so that they do not collapse. You could also just say that they're strong to keep the tube open. And lastly, you need to talk about lignin. So lignin is present and it makes those walls waterproof. Mark yourself out of 2. Next up, we have a lovely set of multiple choice questions. So we're going to start up in the top left. A nucleus will be found in which type of cell? We have xylem and sclenchyma fibers. What property do both of these share? And lastly, we have a bit more to think about here. The cell wall forms a greater percentage of the total mass of the sclenchyma fiber than of the phloem sieve tube. So if you weighed up both of them, the cell wall would make up more of sclerenchyma than phloem. Why? So we have options here. We have phloem sieve tubes do not contain organelles. Phloem sieve tubes have thinner lignified walls. Sclerenchyma fibers contain organelles, and sclerenchyma fibers have thickened lignified cell walls. Tick one of each and we'll see where we go. So let's start off. Firstly, what we need to do is filter out not just what is true and what is false out of our answers, but which of these match the question, and it's about the cell wall forming a greater percentage of the total mass. So, which has the thickest cell wall? What would explain why the cell wall is thicker? So, even though phloem sieve tubes do not contain organelles, this is true, they just have cytoplasm, that isn't linked in any way to the cell wall, so we're going to leave that where it is. The next option, phloem sieve tubes have thinner lignified walls. Well, phloem doesn't contain lignin, so that's nope. Next up, sclerenchyma fibers contain organelles, which is just false. So the only answer that leaves us with is D. Sclerenchyma fibers have thickened lignified cell walls. Next up, we're going to look at our nucleus question. So where will we find a nucleus? Well, a companion cell is our correct answer, because all of these other ones, from all the way down here, even though xylem and sclerenchyma are dead and therefore do not contain organelles, sieve tubes are living but without organelles because they're kept alive by the actions of their companion cell, which provides them with all the nutrients they need. And lastly, this one should be easiest for you after that 4 marker. Xylem and sclerenchyma both indeed provide support, because even though these ones are false, so they don't transport glucose, they don't transport hormones, only xylem transports mineral ions, not sclerenchyma. After here, I have a bit of a drawing task for you. What we would normally do is set up a microscope, we'd look at a few different slides, or we take slides and prepare our own from different pieces of plant tissue, and we would be able to produce our own drawings of vascular bundles. But instead we're going to have to make do. So I want you to look at the 2 pictures that I give you on the next slide. And on some paper, a plain A4 white paper, I want you to draw a nice big diagram, please. Take your time with it. Label up the xylem, phloem and sclerenchyma, and then I want you to annotate each label with the functions of what goes on. So add a little bit about their structure, add a bit about their function. And what we're going to do is keep these for your lab books when we return. So here is our pictures. Feel free to take a screen grab and or just pause the video and draw along. Choose one, not both of them. And then label up the different vascular bundle features and say something about their structure and function. When you've finished, we can check over if you've labelled them in the right place, but I want you to give it a go first. Okay, so next up we are going to have a look at where our labels should be. So let's look at the one on the left first. We have this layer on top, and you notice that it's got very thick lignified cell walls, and yet it's smaller than the one at the bottom, so this has to be your sclerenchyma. Underneath sclerenchyma we know we have the phloem, and the last one has to be the xylem. If you chose a diagram on the right hand side looking at that vascular bundle over here, we're going to look at the different zones again. This time, starting at the bottom, we have those smaller cells with a very thick lignified cell wall, followed by some that don't seem to have a lot of lignin or any, and then you have these cells here that again have that thickened cell wall but look a lot bigger than the ones underneath. So again, we have xylem on the top this time just because of the position of our picture, phloem underneath, and sclerenchyma at the end. So add some labels to these if you haven't already, add your descriptions of structure and functions, and please keep your drawing somewhere safe so that when we come back after the crisis is over, we can put some pictures in our lab books. Thank you for listening along, folks, and feel free to send me an email if you're in my class and need a bit of help. See you later!